to another meeting of the NF-Core Animal Genomic group. As you know, we run monthly meetings, and all of these meetings are recorded. So in case you want to go back to this meeting, because some people normally ask me if they can record, we will post it in YouTube, in the NF-Core channel, so you will have it available. Also, just remind you that you can join us in the NF-Core Slack in the Animal Genomic channel, in case you are interested. And without further ado, I just pass to Francesca, who will present our speaker today. Thank you.
Thank you so much, Jose, and good afternoon, everyone. It's a real pleasure to introduce today's speaker, Dr. Daniela Lourenco. She is an associate professor in animal breeding, genetics, and genomics at the University of Georgia in the United States. Daniela earned her master and the PhD in animal breeding and genetics from the Maringa State University in Brazil. I hope I pronounced it right. She later moved to Georgia for her doctoral collaboration and has been making a global impact in livestock genomics ever since. Today, she leads one of the most influential research groups in applied animal genomics. Her work focuses on using genomic information to accelerate genetic progress, develop statistical methods for genomic evaluations, and designing computational algorithms capable of handling the vast amounts of data generated in modern breeding programs. Her groups has contributed to the implementation of single-step genomic evaluation for beef and dairy cattle, but also swine, poultry, and fishes. And the algorithms and software that have been developed in their group and by our lab are now used for the genomic evaluation worldwide. So it's a true example of research that bridges academia and industry. And the combination of the deep knowledge of quantitative genetics with the hands-on experience in software and methods development shows how that she applied, shows us how genomic selection can become more accurate and efficient, as well as accessible across species. Today, she will show us some insights on this journey, from methods to implementation, and I'm sure we will leave with new ideas on how our community can contribute to the future of animal breeding and genomics. So just a reminder, as Jose said, please note that the talk will be recorded and made available on YouTube. So if you lose it or lose any one of our talks, you will find it on our YouTube channel. And without further ado, please join me in giving a warm welcome to Dr. Daniela Lourenco. Daniela, you can start.
So thank you, Francesca and Jose, for the invitation. It's a pleasure to be here and talk today. And the title that I chose for my talk is What It Takes to Deal with the Largest Livestock Datasets in the World. And I'm going to be straight to the point here. So what it takes a team of dedicated people. So our team right now has about like 30 people. So we are two professors. So there is me here, and then Jorge is an assistant professor. We have 3 researchers, Matias, Fernando, and Andrea. We have 6 postdocs. We have 11 graduate students, 10 PhD students, and 1 master student. And we have several visitors. We also have Ignacy Mistal and Shogo Tsuruta. They started this program here at UGA, but they retired already, but we rehired them. So they're still in the group working like part-time. And we have some external collaborators here as Alberto Cesarani, he's from Italy, and he's adjunct professor in our group. And we have Andres Legara. He's also adjunct professor in our group, and he lives in France but works at CDCB here in the US. And we have other external collaborators as well. So it takes a team of people that work 24/7 to make sure that everything's going smooth. And it also takes collaborators who trust our research and fund our research. So if it was not for our collaborators, we would not have access to the data sets that we have, and we would not have money to fund our research program. So we are very grateful for their collaboration and their contribution to our group. So the work that we do in animal breeding and genetic relies on data. So we have been recording data for like decades, right? So let me give some history here. So in 1895, USDA started collecting data on milk and fat production. And after 40 years, 1935, this is how the DHI data room looked like. So DHI is the Dairy Herd Improvement. So those are the records. When I saw this picture here, I was thinking, okay, those hard drives were really big, right? But actually, they were not hard drives. They were just file cabinets. And then we had here ladies writing all the records by hand, and usually probably they used ladies to hire ladies because the handwriting is better, right? So we had ladies recording all data here. And then fast forwarding to 2023, we can see the amount of dairy data available here in the US. So if we think about yield traits like milk, fat, and protein, there are over 100 million records since 1960s. If we think like cow livability, 121 million records, but some traits that are new, like residual feed intake, we have, this is from 2023, right? About 10,000 records. And what about animals in the pedigree? So the animals in the pedigree, there are about 95 million, and they've been recorded since 1900s. So we can see that there are lots of records, lots of phenotypes, right? Lots of pedigrees, animals in the pedigree. But what about genomic information? Because genomic information is now like a must-have in animal breeding programs. So if you think about dairy in the US, last December, they announced that they had about 10 million genotyped individuals in their database, and they've been genotyping individuals for about 16 years or 17 years now, right? And then if we think about this amount of data, right, 10 million genotyped individuals, I think only CDCB, or not CDCB, but the dairy cattle industry in the US, they are just behind Ancestry DNA and also 23andMe. So Ancestry DNA may have like about 18 million or 20 million samples, and 23andMe may have about 15 million samples, but those are for humans, right? So we are just behind the human data right now. But if we think about 23andMe, only about 5 million individuals are used for research. So records or genotypes on 5 million individuals are being used for research. And 3 years ago, we did this research where we used about 4 million genotyped animals for genomic evaluation. So who did this study? This was done by Alberto Cesarani. So Alberto used 4 million genotyped individuals, 30 million animals in the pedigree, and records for 45 million cows. And then the objective here was to perform genomic evaluation, so to compute genomic breeding values. And for that, we used 5 different breeds just to make things more complicated. We had 3 traits in the model: milk, fat, and protein, and about or more, a little bit over 200 million equations to solve, which is a lot of equations, right? And which method did we use for that? Well, we use single-step GBLUP. And the question is, did it work? Yes, it worked. It took about 72 hours to get genomic breeding values and about 1 terabyte of memory. So it took a lot of memory, right? And for this analysis, we use single-step GBLUP. But what is single-step GBLUP? Single-step GBLUP is a method that was developed here at UGA, and also there was a group in Denmark developing it in parallel. So, but basically, we developed this method here at UGA, and single-step is a method that combines pedigree, phenotypes, and genotypes for all animals available in the data set. So we don't need to have genotypes in all individuals, and this is not a reality in livestock data sets. So in livestock data sets, about like 5 to 10 or 15% of the animals in the pedigree are genotyped. So single step fits this type of data really well because not all animals must be genotyped, right? So it's being like the method of choice for genomic evaluations in animals. It started being used in plants, and believe it your believe you or not, this method also was tested in humans, not for calculating breeding values because we don't call it breeding values in humans, but we call polygenic risk score. So it was tested for computing polygenic risk score in biobank data, so with a lot of genotyped individuals. And for the ones who like equations, so this is the single step GBLUT equation here. So we have the mixed model equations, and here is what we want to estimate. So we'll estimate fixed effects and also genomic breeding values at the same time. And here is our H inverse. So what is this H inverse? It's a matrix that combines pedigree relationships, here represented by A inverse, and genomic relationships. And we also have pedigree relationships for genotyped individuals. And this is just to avoid double counting because we have relationships here, we have here, and we also have here. So what happens in single steps that this H inverse is able to combine pedigree relationships and genomic relationships. For the individuals that have genomic information, we use genomic relationships. For the ones that do not have genomic information, we have pedigree relationships. So, and we can see here that to be able to calculate GBVs, we need the inverse of this matrix, right? That is just composed by 3 other inverses. So just remember this information. And single steps actually under development for over 15 years at UGA and also other places, that as I told you. Actually, it's being under development for 17 years now. And we have already done extensive troubleshooting. So all problems that we identified were solved, and we have developed a lot on the single step methodology. And we have also developed software, and the software suite that we developed is called BLUPF90. And if you want to have more information about BLUPF90, we have this website here, and then you can just go there. We have software without restrictions that can be used in any data set, and this is just under agreement. So those software are not available on the website. But on the website, we have free software for up to 25,000 genotyped individuals, and this is available for research purposes. And I have here some statistics just to show how many access we have in the website per month. So this was, I think, from March, and over 1 million access to the website and about 4,000 true binary downloads. And here we can see just a graph with which software is mostly downloaded. And you can see here in the x-axis that we have a lot of software. So now that you know a little bit more about BLUPF90 and also single step, let's go back to our 4 million genotyped animals evaluation. Excuse me. So I told you that Alberto used 4 million genotyped animals, 30 million animals in the pedigree, and 45 million records, right? Or 45 million cows with records. So if we have 4 million genotyped animals and we want to construct a genomic relationship matrix, right? So based on Van Reyden's approach. So in this case here, this matrix will have a size of 4 million by 4 million, right? So let's do a memory exercise here. How much memory do we need if we want to keep G in memory, right? So if we want to know that, we need to multiply the number of rows of this matrix by the number of columns by 8, divided by 1024 to the power of 4 to get the result in terabyte. When we do that, we see that we need about 116 terabytes just to keep G in memory. And I need to tell you that the servers that we have here in the group have at most 1.5 tera. So having this matrix in memory is not feasible, right? So this matrix, this G matrix is actually very dense, so which means that we have very few zero elements. Most of the elements are non-zero. And so although it's dense, this matrix has a particularity. And to understand this particularity, we need to look at some old concepts. And what are those concepts? So in 1949, Fisher, he came up with this theory of junctions. So based on this theory, Fisher said that at each point of the genome, the DNA is copied from some founder chromosomes. And the junctions are just the points where the founder chromosome of origin changes, right? And then in 1980, Stam, he published this paper saying, okay, we can try to estimate the number of those chromosome segments that are in the genome. And we can try to estimate that based on this formula that is 4NEL, where NE is the effective population size and the L is the length of the genome in Morgans, which means that the expected number of chromosome segments depends on the effective population size. And we know that if we have a small effective population size, we have fewer chromosome segments, but those chromosome segments are longer. And if we have a large effective population size, we have many, many segments, but those segments, they're shorter. So the fact that the number of independent chromosome segments rely on the effective population size, it tells us that there is a limited dimensionality of the genomic information. So there is a limited number of segments, which means there is a limited dimensionality, right? So because of that, the genome of one individual may be just a combination of the segments present in the population. So for sure, there is crossing over that will create new segments, but the most common is that, okay, we have a number of segments, and the new individuals will be combinations of those segments. So there is redundancy here, right? And then we have a system that has redundant information and has a big dimensionality. How do we reduce the dimensionality of this system? To reduce the dimensionality of a system, we can just apply the eigenvalue decomposition. So if we have G and we want to reduce the dimension of G, we can apply the eigenvalue decomposition. But to be more efficient, we can just do the singular value decomposition of our Z matrix. And our Z matrix here is just a matrix of SNP content. So we have animals in the rows and SNPs in the columns. So if we have our Z represented here, and the pink dots are our animals, and the green dots are the SNPs, if we do the singular value decomposition, the blue dots here are just a representation of the dimension that contribute actual information. So the actual information is contributed here by those green dots. So this tells us that the singular value decomposition of Z is a good way to create a sparse representation of our Z matrix while eliminating the noise. So it can just eliminate the noise and keep what is real information here. And when we have the singular values, if we square the singular values, we have the eigenvalues, right? So if we square singular values, we have the eigenvalues. But the question here is, what is the number of eigenvalues of G that would allow us to represent the matrix with all important information, but without the noise, while eliminating the noise, right? About 10 years ago, we had this study conducted by Ivan Pokrinik. So what Ivan did, so he simulated some populations with different effective population size, and he looked at the number of eigenvalues explaining 90% of the variance, 95, 98, and 99. And in this study, we found out that the number of eigenvalues explaining 90% of the variance of G was NEL, 95% was 2NEL, and 98% was 4NEL. And 4NEL is the number of independent chromosome segments that Stam derived in his paper. So here we can say that the dimensionality of G can be approached as the number of eigenvalues explaining 98% of the variance of G. So the number of eigenvalues explaining 98% of the variance of G gives us the number of independent chromosome segments, right? So the most important genomic information in a population is actually contained in a number of individuals corresponding to the number of eigenvalues explaining 98% of the variance of the genomic relationship matrix. So based on that, we developed this algorithm called algorithm for proven and young. And this algorithm actually allowed us to compute G inverse without having to invert G. Just for you to remember here, I told you to just to keep G in memory, we would need 116 tera of memory, right? And inverting a matrix has cubic cost with the number of genotyped individuals here in the case of G, right? So it would be impossible to create this G inverse or to construct G and invert G for 4 million genotyped individuals, right? As we did. So because of the limited dimensionality, we figure out that we could create G inverse without having to invert. And this is what we call G inverse APY, algorithm for proven and young. So in this case, we split the genotyped animals into two sets. One set is the core animals, which is the number of eigenvalues explaining 98% of the variance. So let's say that we have 15,000, as in the case of cattle, right? 15,000. So 15,000 is the number of eigenvalues explaining 98% of the variance of G. So we just select 15,000 animals. Those are the core animals. And then what we do here, we condition the breeding values of all other genotyped animals on the breeding values of core animals. So we do recursions based on core animals. Although we have here a matrix, in the case of 4 million genotyped animals, that is 4 million by 4 million, we just need actually to invert a small portion, which is for the core animals, and all other coefficients of this matrix we obtain based on matrix multiplications. And here, as we condition just on the core animals, we don't have relationships among non-core animals. We just have diagonals for them. And as we have this structure here, we can easily separate this matrix into three portions. This dark blue is for the core animals. This rectangle here is the relationships between core and non-core. And then this vector here is just a vector of the diagonals for the non-core individuals. And because of that, we have a very sparse system, and the computations are very efficient because we can do the computations with those three blocks separately. And if we compare here with the G inverse is very dense because what we do, we condition the breeding value of one genotyped individual on the breeding values of all other genotyped individuals. So we can say here that APY, this algorithm allowed us to construct G inverse without having to invert this matrix. So we can have very efficient computations. So single step now can be very efficient with APY. So here's just the formula that we use for APY. As I told you, we just invert a G for the core animals. All other coefficients is just matrix multiplication. And then, but if we look at the age inverse that we need for single step, we still need A inverse and we need A to 2 inverse. A inverse can be easily constructed based on Henderson-Quast algorithm. A to 2 inverse also, we show that Henderson algorithm applies. In 2017, we showed that Henderson algorithm also applies to compute the A to 2 inverse. So this is just inverse based on partitions of a bigger matrix. And then, okay, so if we go back to our single step with 4 million genotyped individuals, so what we did here, we used 45,000 core animals. And why is that? Because we need to look at the dimensionality within each breed. So as we use 5 breeds here, we had to calculate the number of eigenvalues explaining 98% of the variance within each breed. And this was 15,000 for Jerseys, 15,000 for Holsteins, 5,000 for Guernseys, 5,000 for Brown Swiss, 5,000 for Ayrshires. And then we did single breed versus multi-breed, so 5 breeds. And this, what I'm showing here is the accuracy for cows. This is for protein production. So in blue here, we have the accuracy for single breed analysis. And here in the x-axis, we have the breeds, and we have the number of validation cows per breed. And then when we combined the 5 breeds together, we had at least the same accuracy as for the single breed for most of the breeds, right? So, which means that single step with 4 million genotyped animals is working, right? And we can combine those 5 breeds together. But then I told you that we used about 1 tera of memory and we were not happy with that. So what we did later, so Matthias, who is a researcher in the group, he implemented something that it's called memory mapping. So memory mapping just assigns memory to disk space. It's like what it does, it uses like a segment of virtual memory that has an assigned direct byte for byte correlation with some portion of a file. So we are not using the real memory here, right? So before, what we were doing, we were keeping the genomic relationship matrix in memory, and then we were using the pedigree and the data in iteration on data. So we would read from a file and then do iteration on data. So we were not keeping that in memory. Then we were doing that because before the genomic information was small compared to the pedigree and the phenotypes, but now the genomic information is pretty big compared to the pedigree and the phenotypes. So we did the opposite now. So right now we keep G inverse APY in a disk, and then we keep phenotypes and pedigree in memory. And then when we need to access the G inverse APY, we do the memory mapping. So it reduced the amount of memory considerably. So just one example that we tested here in another evaluation that was taking 720 gigas of memory. With memory mapping, it's actually taking 120 gigas of memory because we were not keeping the G inverse APY in memory anymore, just phenotypes and pedigree. So this allowed us to calculate GBVs with low, for very large data sets with low computing cost, right? So we can have GBVs with low computing cost. And we know that GBVs are published with a measure of accuracy, and it's very hard to approximate that in large genomic data sets. And we had been working with that for like many years, and finally, 2 years ago or 3 years ago, we found a way to deal with that. So we are also using the G inverse APY structure, and what we do, we get information from pedigree and phenotypes, we transform that into weights, we add the weights to the diagonal of G inverse APY, and we do block sparse inversion. So we do inversion separately for non-core and for core animals. And based on that, we can have approximated PEVs, or prediction error variance. So we are able to calculate accuracies, which is used as a measure of precision of breeding values. So right now we can have genomic evaluations for very large populations, and we have commercial applications of API single step, and some companies, they run weekly or even daily genomic evaluations with very large data sets. This is using single step GBLUP, right? But I need to tell you that there are other options as well. The group from Finland, they have developed what we call single step GT-BLUP or GTA BLUP right now. So these use Woodbury inverse. And there is also single step SNP BLUP. So single step SNP BLUP is calculates the SNP effects. So it has the largest dimension is actually the number of SNPs by number of SNPs, right? So, and the choice of method, if you're using single step GBLUP, single step GT BLUP, or single step SNP BLUP, is based on what you like most and the one that will take less memory and less computing time. But the good news here is that all 3 methods are equivalent. And the fact that the methods are equivalent is a good thing. And why is that? Because if you think about, let's say, single step GBLUP and single step SNP BLUP, right? So they're equivalent models under same assumption and data, which means that if we have SNP effects from SNP BLUP, we can calculate GBVs here. Using this formula here, we just multiply the SNP effects by a matrix of SNP content. And if we have breeding values, genomic breeding values from single step, we can back solve it using this regression here to get SNP effect. And also, we can get the variance of SNP effects, right, using this formula here. And the variance of SNP effects can be used for what? To calculate p-values for SNPs. So if we get this variance of SNP effects, we take square root. This is the standard deviation here of SNP effect. And here the SNP effect itself, the estimated SNP effect. And we apply to this formula and we can get p-values for GWAS. So see, the equivalence allows us to do multiple things. And then we have also implemented single-step GWAS. So to take into consideration that the livestock data sets, we may not have all genotyped individuals, right? So we can account for all the information if we have single-step GWAS. And what if we are using APY, single-step GBLUP? So if we're using APY and single-step GBLUP, we also have an equivalent APY single-step SNP-BLUP model. And this equivalence is very good. And why is that? Because we have derived some formulas and we show that if we are using single-step, to calculate the SNP effects, we only need the G inverse for core animals, GBVs for core animals, and the Z matrix for core animals. And the SNP effects are equivalent here. So, and the variance as well, we can calculate the variance of SNP effects only based on information for core animals. Although we have the full evaluation with all genotyped individuals, we just extract coefficients for the core animals and we apply to those formulas and we can get SNP effects and also variance of SNP effects. So everything can be put in function of core animals. And because of that, we now can have single-step GWAS unlimited. So we can calculate p-values for SNPs in very, very large data sets with millions of genotyped individuals. And how do we do that? Well, we have the GBVs from the genomic evaluation process. We also have the accuracies using the block parsing version. And this here is what we need from the accuracy approximation. So we extract this portion from the accuracy approximation and we can get our p-values, right? So this is from the blocks parsing version that I showed you before. And then we have done some applications here. So Natalia Leite, he was a PhD student here in the group. So she compared the traditional single-step GWAS with 50,000 genotyped individuals with the single-step GWAS extended to large genotyped populations using 500,000 genotyped animals. And we saw here that the resolution of GWAS was better when she used more genotyped individuals. So the sample size really matters for the resolution of GWAS. And then if you ask us how single-step GWAS compares to methods like Emacs, right, that is being like widely used, especially in human populations. So we simulated some populations here with 1 QTN per chromosome. And then here are the results from Emacs and also single-step GWAS. We can see that the results are very similar, but sometimes we have a little bit better resolution here because in single-step we can use all the information available. And in Emacs, we had to use only genotyped individuals and calculate the regress EBVs for those genotyped individuals. But if we compare the statistics like true positive and false positive, there were no difference between Emacs and single-step GWAS. And then talking still about single-step GWAS, I want to show another application here. So this was for fat yield. And Eula, she was a postdoc here in the group, and then she worked with this data set. This is from CDCB. So she used about 2.35 million genotyped individuals from all breeds, pedigree for 52.7 million animals, and phenotypes for 107 million animals. And this is the Manhattan plot for fat yield. So we have here the minus log 10 of p-value. We can see some peaks here. And the QQ plot also looks good. So if we compare that, right, the Manhattan plot from Eula's study with one study that I did like 6 years ago with about 10,000 genotyped individuals, we can see here that, okay, we see a peak on chromosome 14, and this SNP here is DGAT1, and the minus log 10 of p-value was about 12. For Eula's study, this here also is DGAT1, and the minus log 10 of p-value is about 100. And also here we can see one peak here on Eula's Manhattan plot, where on chromosome 5, where we see a significant SNP, but in my study, this SNP was not significant. So the amount of data in GWAS really helps us to find significant SNPs. It really helps us with the resolution, right? To improve the resolution of GWAS. And we did some studies recently to try to understand this relationship between effective population size, a number of independent chromosome segments, and sample size for GWAS. And this was done by Sambong. Sambong was a PhD student in the group. So he simulated the population with 200 effective population size, 30,000 genotyped individuals, 2,000 QTNs, and the effective population size was 15,000. Because sometimes also people ask, what is the ideal sample size in GWAS, right? Our best guess would be the number of independent chromosome segments, which is equivalent to the number of core animals in APY, right? So in this case here, Sambong, he did the GWAS using 15,000 genotyped individuals, and each individual had a single phenotype. And then we can see here that about 7 peaks were significant, but he simulated 2,000. What happened with the other peaks? So we decided to increase the number of genotyped individuals to 30,000, the maximum that we simulated, and we could double the number of significant peaks, so about 15, but still we simulated 2,000, right? So what we decided to do was to increase the sample size, or not to increase the sample size. The number of genotyped individuals was still the same, but we increased the amount of information for each individual. And what is the rationale on that? So if we have a lot of information for genotyped individuals, this means that the accuracy of GBVs are pretty high, are close to 1, right? If the accuracies are pretty high, so if the breeding values are pretty accurate, the SNP effects will also be accurate. So the error in estimating SNP effects will be smaller, so which means that we can find more significant SNPs. So, and this is what we found here, 15,000 genotyped individuals, but each individual had lots of progeny records, and we could find many peaks, right? And then we tested as well in 30,000 genotyped individuals, and we could see many peaks, but it's not very different from what we found with 15,000 genotyped animals. So this tells us that the amount of data that we use for genotyped individuals is crucial if we want to have a good resolution and find the QTNs in the population. So using more data allows us to have less noise and higher resolution. So this was true in a population with about effective size of 200. But what about in smaller populations? Because if we think about like pig populations, chicken populations, the effective population size is about like from 20 to 40, something like that. In cattle populations, the effective size can be like 100, 150, 200, right? So in a population with a small effective size, we were not able to find many QTNs here, even after adding lots of progeny records. And why is that? If we look back at our segments and the dependence of segments on the effective population size, if we have small effective population size, we have larger segments. So it's really hard to pinpoint the correct QTN if we have lots of SNPs trying to steal the signal of the QTN here. If we have large effective population size, we have shorter blocks, so less SNPs trying to steal the signal of the QTN. And to prove that, we had another study where we simulated some chromosomes and SNPs in exactly the same place in the chromosomes. The SNPs were exactly in the same place, so we could average their effect. Excuse me. And then in this case here, the first case, we simulated a population with effective size of 60, and the number of genotyped animals with phenotypes was 6,000. So here we had the QTN here in red with effect 4, And then we could find one SNP here with effect about 2.8, but we can see here that lots of SNPs are trying to steal the signal of this QTN. Then we increase the sample size to 18,000, so 18,000 genotyped individuals with phenotypes, still the same effective population size, and we could see here that the effect of the SNP was a little bit higher, about 3.5, and we had lots of SNPs trying to steal the signal. When we increase the sample size, the effective population size to 600, the sample size was still 6,000, right? But the effective population size was 10 times bigger than before. We saw a stronger signal here, stronger effect, and the SNPs are not trying to steal the signal. So here we have the case of longer LD blocks. Here we have the case of shorter LD blocks. And then this showed us that, yes, effective population size plays a role on the resolution of GWAS as well. And if you want to see more about this study, we published last year, and it's this paper here. So we do need to, if we have a larger effect, or if we have a smaller effective population size, we need a lot of samples to be able to have a higher resolution in GWAS. So sample size is extremely important. But what about the number of SNPs? Is the number of SNPs important if we're thinking about having more accurate breeding values? Well, for some time now, we've been thinking about sequencing, using sequencing data, right? So should we use sequence data for genomic predictions? So we know that sequencing became very cheap. Just to give you an example, if the price of this Ferrari had dropped in the same way that the price of sequencing dropped, we would be able to buy this Ferrari for about 1 cent of dollar, right? So sequencing is very cheap right now. So we joined a project with PIC and Roslin to figure out if we should use sequence data for genomic predictions. And then we had this data set with some genotyped individuals for several lines, maternal and terminal lines. Then the guys from Roslin, they sequenced some individuals, like 1-2% of the individuals in the population. And then all those individuals had imputed sequence. So after sequencing, the imputation was done to sequence. So this is the number of individuals with sequence information. So at the end of the day, we had almost 400,000 individuals with sequence. But we did not use all the 10 million to 20 million SNPs. We pre-selected SNPs, and we added those pre-selected SNPs to the SNP chip that PIC uses. And so in blue, we have here the accuracy, the prediction accuracy for the chip, in green for the chip plus the significant SNPs that were selected from sequence information. So we can see here that for all the lines, the increase in accuracy was very small. So maximum 0.01 when we used sequence information. So maybe using sequence information is not useful here. So we can say that genomic selection has been very effective with the number of SNPs that we have been using, like let's say 50,000 SNPs, 60,000 SNPs, right? So genomic selection has been effective with the number of SNPs that we have right now. So talking about genomic selection, I just want to touch quickly on something here that is, it seems that we are having some winners and losers of the genomic selection game, right? So we have seen some informal reports where in some species there are some traits that are being affected negatively, right? So if we think about pigs, winners may be like growth, production, litter size, losers, sow survival, pelvic organ prolapse, and new diseases that are happening now that were not common before. Excuse me. If we think about dairy cattle, winners, fat production, protein production, productive life. Losers may be calf mortality that is increasing. The cows are having shorter teeth and they are less tolerant to heat. If you think about beef cattle, winners may be growth, production, and meat quality, and losers may be feet and leg structure, congestive heart failure for feedlot animals, that it's happening a lot, If we think about chickens, winners are growth, meat percentage, and losers may be number of eggs and also fertility. But then how can we investigate if there are real losers in the genomic selection game, if those traits or those diseases or those traits that are detrimental are happening more often here because of genomic selection, right? So how to rule genomic selection out or in these those problems that we are observing, right? To do that, we need to be able to estimate variance components and also genetic parameters like correlations and heritabilities over time. And we should be able to use as much data as possible and also genomic information, because we have seen that if variance components, if the selection in C includes genomic information, but variance components estimation does not include genomic information, we may have biased variance components. So we should be able to use genomic information, a lot of genomic information, to be able to correctly estimate genetic parameters and variance components. But we have limitations here. With large data sets, it's extremely impossible to estimate variance components. If we have like many genotyped individuals, this is not going to work, right? So we do need software optimizations and new methods. So we have been working on that. And Matthias recently did some software optimization on Blood F90 Plus. And this, so instead of using a sparse matrix package called YAMS that requires a lot of time here, we are using some dense matrix computations from MKL that is extremely efficient. And we have seen that the computing time is really small here compared to using YAMS. But we were able to do that for about 60,000 genotyped animals in very simple models. After that, over that, we cannot do it. Well, actually, we could not do it because we have this paper that it's accepted now. It will be out soon, where we implemented what is called Monte Carlo Remo, that combines also simulation of GBVs from single-step GBLUB. So this method allows us to estimate various components with very large data sets, and we are calling it MC single-step GREMO. So we tested that for some populations. So the first population we tested in a small population, so we were able to compare this MC single-step GREMO with the regular single-step GREMO. So we were able to use about 10,000 genotyped individuals for a 3-trait model. And then single-step GREMO took about 6.5 days and 105 gigas of memory. MC single-step GREMO took 1 gig of memory and 12 hours for this small data set, so it works. And then we extended that to about 331,000 genotyped individuals and 3-trait model. And we were able to get estimates in 5 days, which is pretty reasonable for very large data sets, especially with genomic information, with many genotyped individuals, that this was not possible before. And we are also testing a different method that we call now GPP, genetic parameters via predictivity. So this is just like Ignacy derived some formulas here, and we are testing right now to estimate heritability and genetic correlations just based on predictive ability formulas. So we are, as I told you, we are still testing it with simulated data, but for traits with high heritability or moderate to high heritability, we have seen that it's working pretty well. So this is something that we are still investigating, but at least we have one method that it's working very well. And then things that we need to be concerned right now is the extra source of information that are becoming available, right? So we have projects here where we are trying to include extra source of information in genomic evaluation methods. So one project is with environmics, so to better understand the environment and include that information in genomic prediction models. Another project that we have is with intermediate omics data, in this case metabolites. So we are trying to include metabolite information in genomic prediction models. We have the first paper ready to submit on this one, and this project is led by Hao Sheng from UC Davis. And then another thing that we are also concerned now is digital phenotyping, right? So we can see that many features can be extracted from videos here. I think this video is not working, but anyways. And then we can extract traditional and also novel phenotypes. But the question is how to deal with digital phenotypes, right? Because the amount of information that we get from videos, it's overwhelming. And some companies, they have over 100,000 individuals with image data, right? So with videos. So right now we've been working hard to try to understand that and try to extract useful phenotypes and try to use those useful phenotypes in genomic prediction models. So we can see right now that the amount of data that we are collecting and that we are trying to use in genomic prediction models is extremely large, and this brings a lot of challenges. But I can tell you that challenges are good because it kind of guides us to next development, right? So although we have several challenges, we can say that having good collaborators like companies and associations that are willing to share data and also to finance, to fund our research and have lots of people with different backgrounds is extremely important in this scenario, right? We need to have people that have statistical background in animal science, in biology, computer science. So those are the backgrounds of people that we have here in the group. So with that, we are able to develop new methods to deal with new source of informations. We are able to do extensive testing because some people may be better in developing methods, some people may be better in testing the methods, some people may be better in writing software. So we need to have this very diverse background. We can say that regarding large data for animal breeding genetics, we can have genomic evaluations like having GBVs and accuracies in a reasonable time and using a reasonable amount of memory. We can also have single-step duals with p-values for SNPs. We can have variance components estimation for very large genomic data sets. And right now, the things that we are working on, as I told you, is to include other omics. So this still needs some attention, but yeah, there are several groups working on that. And also, we need always to be aware of the needs of the industry, because if we don't know what they need, we don't know what to develop. So, and we need to be able to develop new methods and tools on time, right? And what are the important skills that we need to develop that? So, we need to be able to program in several languages. This is extremely important for grad students. I always tell the grad students that they need to take advantage of this time to learn as much as they can, right? And there are several very good skills that they need, and one of them is programming, right? So although like you can learn one language, if you learn one language, you are able to program in several languages. The syntax change a little bit, right? But if you know the logics, knowing the logics is the most important thing, right? We also need to have a strong understanding of the theory in our field of study, because some people, they know how to program, but they don't know how to code, right? So it's very important to bring those people to the theory so they will understand what they need to program, right? And also knowing the theory allows for new developments and allows for insights on what can work and what cannot work, what we should spend time on or what we should not spend time on, right? Also important skills is to be an independent thinker, right? So what I do with my students, usually like the first year, second year, I help them more. In the third year or half of second year to the third year, I start helping less, because in this case, they're kind of in a winning process, right? Because once they finish PhD, they are going to go to the industry or to academia, and they are not going to be able to call me every day to ask what to do, right? So they need to learn how to be independent thinkers. So being a thinker is very important, but being a doer is even more important, right? So we need to know how to think and how to do things. We need to have soft skills and teamwork as well. Working in teams is extremely important. And the last thing, we need to be confident. So if we are confident, we know that we can be anything that we want to be, right? So I think I'm already like running out of time. So if you have any questions, you can email me. So if you don't have time to ask here, this is my email. And if you want to know more about my group, here is the website. And know more about the programs that we develop, here is also the website. So with that, I'd like to thank you all for being here, and I'll be happy to take questions if you have them.
Okay, thank you so much, Daniela. It was a really beautiful presentation. Do we have some question from the audience? Or otherwise, I will kind of break the ice. Okay, so I have just a curiosity, because you showed us a lot of information and data, and I kind of wanted to tell you, what do you think will be the next big leap in animal genomics over the next, I don't know, 5 to 10 years?
Mm-hmm. Yeah, that's a good question. And it's really hard because I always tell people, so genomics was a big leap, right? So we started using genomics in 2009, and it was a big leap. Why? Because it helped us to increase the accuracy of predictions. And why is that? Because we added a source of information that was non-redundant with the information that we already had in the system, right? So we already had pedigree and phenotypes, and genomics brought some extra source of information that was non-redundant. Right now, we are in a big search for non-redundant source of information, right? As I told you, we are trying to see if a metabolomics data would help, if microbiome data would help, and also soon transcriptomic data would help, right? But there are several layers to those source of information because most of them are time dependent. So we don't know what time and which time they should be collected to help us, right? So, but those are the, like adding omics, I think is the next step. But it's going to be useful only if they add extra information that's non-redundant to the information that we already have, right? And another big leap as well is with the digital phenotypes. And why is that? Because we have several traits that we record that are not so accurate or are not so easy to record. One example is that this project that we worked with PIC. So they record feet score for the animals, front and back leg score actually. So, and this is categorical. So they have technicians recording, and the record by technicians can be different depending on the technician. It's technician dependent, right? So what they did, they recorded that based on videos, and then we use that information, right? So we calculated like heritabilities for those traits based on technicians and based on the digital phenotypes, and the heritabilities are 3 times bigger, which means that there is less noise. And also because the first based on technicians was categorical, and based on digital information is continuous, so it's adding more information here. So I think those are like omics, and also digital phenotypes will add extra information to our evaluation. I think we will go kind of in that direction in the next 5 years at least.
Yeah, I agree with you. So we have time for 1 or 2 questions, right? Like quick ones.
Is there a question in the chat?
Yeah.
What is the difference, if any, between SS-GWAS and SS-SNP?
Okay, so single-step GWAS is actually we are trying to find genes or QTLs associated with the trait, right? So we are seeing if SNPs are statistically significant or statistically associated with the trait, right? This is the GWAS. So single-step SNP-BLUP actually is a prediction model. It's not a genome-wide association method. It's a prediction method that will provide us SNP effects for the individuals that are genotyped and breeding values for the individuals that are not genotyped, right? So 2 different things. Single-step SNP-BLUP is for prediction, and then single-step GWAS is for genome-wide association.
Okay, and we have another question from Simanta, I think, who is asking, how do you see the future of animal breeding research in the time of AI, and how should upcoming research aim to be experts like yourself should prepare for it?
Yeah, I think we should be prepared for it for sure, right? So I think AI will help us in some ways, like in being more effective in studying, finding papers related to some topics that we want, right? And it will also help us to optimize software, right? So we know that those models, they have been optimized for software development. So I think AI will help us with that. But still, we need to be very careful with that because we forget that the machine is trained for something and it cannot see outside of what it's trained right now, right? One example, we have software called PreGSF90 that is used to do quality control of genomic data and also construct the genomic relationship matrices. I asked ChatGPT how to perform cross-validation with PreGSF90. PreGSF90 does not do cross-validation at all, has nothing to do with cross-validation. The answer was, oh yeah, since 2019, PreGSF90 was changed with the capabilities of doing cross-validation. And then to do cross-validation, you need to add this option and this option. Those options did not exist at all, right? So we need to be very careful because sometimes they're very convincing and they give us wrong information. Those models, they give wrong information. One thing that I say is that the machine learning will never come back and tell us, sorry, I did not learn anything. They will try to place something to answer the question that you asked. So we still need to be very careful with that. But we need to know how to use the tools and we need to be prepared.
Yeah. We still have one question and then I think we will close it. Is still from Alexandro, who is asking, can modern deep learning models, for example, transformers, be good contenders for classical methods based on GBLUP when doing genomic prediction?
Yeah, so there are some attempts to use machine learning methods for genomic prediction, but the type of model that we use, they are linear models. So that's why right now, machine learning methods, they are not better than the classical GBLUP or BLUP method for predictions, right? But at some point, we may learn how to use those machine learning methods for genomic predictions. But right now, they are not as good as GBLUP because of the type of models that we use. We use linear models, right? And then some people say, oh, with machine learning methods, we can extract non-additive effects, right? But if we think about genomic prediction, we are interested in additive effects, not in non-additive effects, right? So there are several layers to this question. But anyway, so far, GBLUP or BLUP-based methods are better than machine learning methods for prediction.
Okay, so thank you so much. I will call in Jose to close the session. And then if we still have some question, you can just write to Daniela or even propose them on our Slack channel.
Yes, so yes, thank you for the very nice talk, Daniela. And just wanted to, I just put it in the Slack, but I saw some people joining at the end of the meeting. So just a reminder that all of our past meetings are in our website, in NFCore. You can go there using the link that I have put it there. And we will run the meeting for November, but we have still to confirm the speaker, but we will announce it. And yes, so I hope to see you all of you then in our next meeting. Thank you very much for joining.
Thank you.
Thank you, Daniela.
Thank you. Bye.
Bye-bye.
Bye. Thank you. Did I